Thank you so much. So good day, everyone. I'm really delighted to be here today. And I'll be speaking about maternal immunization, clinical research in low and middle income countries, the study design, ethical, regulatory, and safety considerations. So I worked for, I did my training in one of the largest pediatric hospitals in Asia and really saw firsthand the benefits of maternal immunization because we had these neonates who would often get infectious diseases. There was no treatment available in some of these large public hospitals. And, I really saw firsthand how important maternal immunization is. We lose about 2.3 million neonates in the first month of life. That's approximately 6,500 neonatal deaths per day. And maternal immunization or vaccinating of pregnant women is a promising strategy to reduce the morbidity and mortality in the pregnant woman, the fetus, and infants. So it works in two ways. You could prevent infection in pregnant women with the subsequent prevention of transmission of infection to the infants. And maternal immunization also protects the fetus and infants in the first month of life through placental transfer of the IgG antibodies and or IgA antibodies being transferred through the mother's breast milk to the infants. And maternal immunization is effective before active immunization of infants can be initiated, or the infants can actually respond to infectious challenges. So in many high-income countries and LMICs, we've got vaccines like tetanus, pertussis, seasonal influenza, and COVID-19 vaccines recommended for immunization of pregnant women. And we expect the RSV vaccine for pregnant women to receive a potential approval in August 23. The new maternal immunization vaccines under development, including Group B strep, cytomegalovirus, and malaria. I'm fortunate to be doing quite a bit of work on Group B strep vaccines currently, which I'll come to later. So basically what we're trying to do is with the antibodies being transferred from the mother after she's vaccinated to the infant, we're trying to narrow the window of vulnerability that we're seeing in the early months of the infant's life. Now, coming to the study design and ethical considerations of maternal immunization clinical research in LMICs. So, the first and foremost principle of all, licensure of vaccines is that the vaccine should have a favorable risk-benefit profile in the defined population, and only then are vaccines licensed. And so for all adverse events, the investigators would have to make a judgment on the potential relatedness of any adverse events to vaccination, depending on the possibility of the relatedness, the timing in relation to the dosing of the vaccine, and anticipated background rates. So background rates would be the baseline rates of the maternal and neonatal outcome that we would expect to see in that population. But the issue is that background rates on the disease burden and maternal and neonatal outcomes in pregnancy in the study population are often absent. So we know, for example, that major structural or genetic defects affect about 3% of all births in the US, and they're associated with about 20% of all infant deaths. But if we did not have this background rate available, then any congenital anomaly, for example, that we would see, we could attribute it to the vaccine. So it's critically important that we have these background rates, which are needed for sample size calculation and determination of the actual risk for research participation from the vaccine administration. In LMICs, we've seen that public health records, which are often collected by different agencies at different points of time using different methodologies, are unreliable. And there's a lack of use of standardized case definitions to distinguish conditions like stillbirth from abortions, and there's a lack of accurate gestational age assessment in used in these public records. So it's really useful to determine or validate background rates in the study population prior to the clinical trial. In fact, currently I'm working on background rate determination for key maternal and neonatal outcomes in one of the low-income countries in Africa, and we're doing this in preparation for setting up a pregnancy registry there. So, there's always a risk-adjusted study design. So you would progress from healthy women in a reproductive age group to pregnant women. So you would have an incremental increase in risk as the chances of likely benefit increase for the pregnant women to participate in the trial. So the FDA framework for vaccines for pregnant women proposes that, first non-pregnant women of childbearing potential, a phase 1 and phase 2 trial of the vaccine candidates is done in this population, and then pregnant women are enrolled in later phases of the trial. Similarly, the National Institute of Allergy and Infectious Disease used a tiered approach for the H1N1 vaccination, where we had safety and immunogenicity data from routine vaccination, first collected in the general population, and then clinical trial done in women during the second and third trimester. We typically would like to initially administer vaccines during the second and third trimester because the first trimester is the period of organogenesis. So we try and avoid vaccine administration in the first trimester. That's one of the reasons. And the second reason is that, it's very difficult to get pregnant women to come for antenatal care visits during the first trimester. So this risk-adjusted study design helps to minimize safety concerns and targets the population most at risk of severe disease. In terms of inclusion-exclusion criteria, so gestational age windows are often specified for administration of the vaccine, and they're typically based on the last menstrual period and fundal height. But these are often inaccurate in LMICs. Women might not remember the last menstrual period, fundal height might not be determined accurately. So a recommendation is that we use standardized case definitions for gestational age determination. For example, I've led the development of a companion guide for the assessment of gestational age, which is now available online. And, it's very clearly spells out how gestational age actually should be determined. So, in a standardized methodology, which can be used across different countries. The use of portable, inexpensive ultrasound machines is very important for gestational age assessment. In fact, the study that we're currently doing in the Gambia, we are using these ultrasound machines for gestational age assessment in clinical trials. And it's important to train the research team and quality assurance of scans is to be done. Recruitment of pregnant women in the first trimester is very challenging, because pregnant women often don't want to disclose their pregnancies in LMICs and, early in pregnancy because they're scared that there might be some harm that would befall the fetus. So this limits the ability to capture information on fetal development early in the pregnancy. So our recommendation is to emphasize and provide benefits for early enrollment in antenatal care. We found small measures like providing free supplements to the women, say calcium supplements, or providing them with a picture of their ultrasound scan to take home to their family. That really helps motivate women to come in for the antenatal care early in the pregnancy. There's also lack of trust for the clinical research and lack of autonomy for pregnant women, often in LMICs. It's felt that pregnant women cannot make their own decisions and they need their permission from their spouses or their partners, from the elders in their family, or from community members to take any key decision, including participating in a clinical trial. So a recommendation is that we engage with the social, community, political leaders, and extended family. What we found that really works well is that before we plan any clinical trial in in LMICs, we really help work to educate the community about what it is that we're trying to do. We set up a community advisory board. We work closely with maternal, neonatal, and child health and antenatal care and local healthcare providers, because pregnant women are very familiar with the local midwives and the local healthcare providers, and they trust them and they go to them. So, the community volunteers and midwives, we often brief them on the details of the study, the inclusion exclusion criteria, and the involvement of the study participants and study outcomes. And then this helps to really disseminate the information about the clinical design and what it is that we're hoping to do in the studies. And once this information comes from people that they trust, and the pregnant women are more open to participating in this research. The site can also conduct community meetings around the catchment areas, organized through community leaders or in partnerships with local NGOs working in that field. What we found worked really well also was to use local street theater groups in partnership with local NGOs in some of the studies that we did. to disseminate information about the clinical research and explain what it is that we were trying to do. And that really helped to build up people in the community's general awareness about the research. In terms of recruitment, the researchers often develop very close relationships with community leaders and public health workers. So it's important for the researchers to actually advocate for improvement of public health facilities and local health centers. And this ensures that the community is volunteering to help with the research actually get sustainable access to standards of care. And this helps to build up confidence in the community. And then, we've often had no issues about recruiting for clinical trials in pregnant women in these communities, because there's just so much trust that's been built up with these, in the community. Now, coming to informed consent, the factors that impact consent in LMICs include a low trust for authority. It's ensuring comprehension of the risk is challenging in pregnant women. And we often find that there's withdrawal of consent due to input of family members. And especially, in some of the countries that we work in, there's a preference for, female for male infants. And so, if the family members find out that the woman has a male fetus, and so then they would not want her to participate in the clinical research because they fear that some harm might come to the pregnant woman or the fetus. So it's important to plan for repeated consent discussions with pregnant women. And if desired by the pregnant women, then the researchers can explain the benefits and the risk of participation of the family members and community leaders. And it's very important to provide easily understandable, culturally sensitive, comprehensive information in locally spoken languages on the benefit and risk of maternal immunization, so that the woman really can understand, what it entails participating in the research and can give her informed consent. It's important to invest in staff trained communication skills to promote the understanding of pre-trial concepts, to get community inputs to the informed consent method that are used in the trial, assess the understanding of the pregnant women in a very rigorous way, especially for key concept questions. It's important to reduce the risk of trial procedures to acceptable levels. And all the protocols we submit to the IRBs, we have to really declare all the strategies for recruitment in the protocols. And it's important to have longer timelines for enrollment and, possible high attrition rates, which are planned for by the researchers, because that is the ground reality of what we found in LMICs. In terms of culture-specific considerations, so in case your clinical research involves disclosure of pregnancy in the first trimester or disclosure of the fetal sex, that is very important to be well aware of the culture-specific norms. In case, for some of the countries that we worked in, there's a high rate of female infanticide. We see that in some of the countries in Asia. So then the researchers have to be very careful not to disclose the sex of the fetus, because, women might just be coming to the research center to determine the fetus sex, because it's illegal to disclose it, across the country. Placental sampling or large blood draws from pregnant women or neonates, or stillbirth or autopsy studies might be challenging in some contexts. We've had cases where the community actually thought that, because we were withdrawing blood samples, so then from the pregnant women or the neonate, that the researchers were actually selling the blood samples or using the blood samples for voodoo purposes. So we had to really sit down and explain to the community and to the trial participants why we needed the blood samples, and what were the reasons for all the steps that we were doing in the trial. So it's very important to have a clear understanding of the culture-specific considerations in the country that you're working in. The other thing to think about is that more than 1/3 of young women in LMICs are married before 18 years of age. So you have to consider the implications for consenting adolescent pregnant women if you plan to enroll them in your study. In terms of research data collection, the deliveries are not often conducted in health facilities identified by researchers. And often you would have a single midwife or healthcare provider for multiple women in labor, in maternity wards, especially in government hospitals. So there would be limited assessment of the woman or infant done at discharge, including limited information on the birth weight, head circumference, height of the of the neonate, and because the healthcare providers are so overwhelmed that this data is just not available, and it's difficult to link the maternal and infant records. So you have to do really detailed logistic planning, intensive training of the healthcare providers, to explain what is the importance of collecting the data and why the accurate data collection is so critical. Quality assurance has to be done, and quality control measures have to be consistently done throughout the trial for data consistency and integrity of the data. In terms of standard of care, there's often disparity between the standard of care policies in the country, and excess that pregnant women actually have to care in their countries, because the standard of care might be very good on paper, but the ground realities, especially in public hospitals, might be very different. So there's a need for capacity building in the study communities to meet the standard of care in the country for the pregnant women and neonates. And it's important that active detection and treatment of conditions in pregnant women is done. This could be for anemia, hypertension, malnutrition, infectious diseases. These are all conditions we see very commonly among pregnant women in LMICs. And, so often we would provide treatment for these conditions during the course of the trial. And so this would impact on the maternal and neonatal and infant outcomes. So the outcomes actually would be much better in the course of the trial, because the women have actually received very good care. So this would help to decrease the background rates of key outcome measures in that particular population. And so the researchers would have to factor in the decrease in these rates of outcome in the sample size calculation. Say if you're looking at preterm birth and the pregnant woman has received good care for her hypertension or for her diabetes or for her malnutrition, the rates of preterm birth in that study would decrease. And so then you need a larger sample size in that study to be able to reach your end goal of your certain number of preterm births occurring. In terms of follow-up, long-term studies of child development and women's health may be needed based on the vaccine or adjuvant properties. So a practical implementable follow-up duration in LMIC should be considered. No sense in saying that you're going to follow up the neonates for 2 years, because that's never going to be practically feasible. You have to really make sure that whatever you're mentioning is actually practically feasible in that setting. So the minimum recommended follow-up period for women could be 6 months post-delivery or following early termination of pregnancy. And the minimum recommended follow-up period for infants could be until 1 year of age. And, extended follow-up could be based on the properties of the vaccine, results of the preclinical studies, depending on the disease that you are looking at, the pregnant women's nutrition or immune compromised conditions, or what is mentioned in the risk management plan that the regulatory authorities then would mandate that, if, say, you're looking at a population with a, pregnant women with HIV/AIDS, they might want you to follow them up for a longer duration of time post-delivery or, post birth of the neonate. There's a risk of reduced adherence to intensive follow-up visit schedules over the course of the study. And in LMIC, there's a tradition of women going to the maternal homes to deliver. So in clinical research, women should be included who agree to deliver at the designated health center. But researchers should plan for delivery plans changing over the course of pregnancy and women returning to their maternal homes for the delivery. That might make it difficult to follow up the woman and the neonate post-delivery if she's gone off to a maternal home, which might be at a distance. So a pregnant women should receive the standard of care prevalent in the country for infectious diseases, non-communicable conditions, and this might actually alter the pregnancy outcomes. You might have better pregnancy outcomes occurring, and this would limit the conclusions about the safety and effectiveness of the vaccine. So this increases the importance of prospective structured pharmacovigilance studies post-vaccine introduction, which would help in the detection of uncommon or delayed onset vaccine reactions occurring. Now, coming to the ethical considerations of maternal immunization research, this is a really exhaustive topic. I'm going to be just touching on a few points in, keeping the time that we have available, and we could discuss this in detail, say, in another presentation, perhaps. So the rationale for maternal immunization research, the older approach was that the disease risk must be concrete and severe for both the mother and infant before maternal immunization is justifiable. But the revised ethical consideration that we should account for the pregnant woman's strong interest in having a healthy rather than sick child would be a better rationale for the maternal immunization research to occur. Now, informed consent. So based on the principles of autonomy, the permission of another person cannot replace the requirement of individual informed consent by pregnant women. And if the pregnant woman desires, then information about the research can be shared with her spouse and extended family. So in one of the studies that we were doing in one of the LMICs number of years ago, we found that the researchers had added in an extra layer. So, the pregnant woman would consent, but then there had to be consent also obtained from her spouse or from her partner to be able for the pregnant woman to enroll in the study. And so then we had to really explain to the researcher that the pregnant woman is an autonomous person. She can make her own decisions, and only if she wants, then the information about the research can be shared with the spouse and extended family. But you cannot take away the ask for additional assent from key decision makers apart from consent from the pregnant woman, because this would disempower the woman from making her own decisions. So she's an autonomous person. She can take her decision to participate in the study. And if she so desires, then additional information can be shared with the spouse, extended family, or community members. But there's no way that you can add an additional layer of consent from another key decision maker on top of the pregnant woman's consent to participate in the study. If you're trying to enroll adolescent pregnant women, then the interesting thing is that the parents would have to give the consent, and the pregnant woman would have to give her assent to participate in the clinical research if they're less than 18 years old. But once the pregnant adolescent delivers, then she can actually give consent for her newborn child to participate in the research. So it's important to evaluate the potential risks and benefits. So for research's potential of direct medical benefit to the woman or fetus, the risk proportionate to the potential benefit is acceptable. But for research with no prospect of direct medical benefit, risk to the fetus must be no more than minimal. And the reason for this is that the fetus or infant cannot consent to participation in the research. And it's difficult to determine how much risk is acceptable to impose upon the fetus or infant. So the definitions of minimal risk in the context of pregnancy are unclear. So we need methods for estimating the incidence rates, testing hypotheses, and determining causal associations of the outcome of maternal immunization to provide evidence of actual risk from participation in the research. So you really need a very well planned research which meets all the regulatory guidelines required in that country. You need to have background rates and very good safety monitoring to make sure that the risks and benefit are favorable for the pregnant woman to participate in the research. In terms of decision making, there's an active dialogue between pregnant women and healthcare providers, which is important to understand the pregnant woman's beliefs about the necessity and safety of maternal immunization and address any concerns. And it's very important to address the concerns of pregnant women so that she's really very clearly informed about the risks and benefits of participating, and then can give consent for participating in the trial based on full knowledge of what it entails. So this active dialogue contributes to ethical justification and trustworthiness of the program. And women should be adequately represented in decision-making bodies that influence the national and international level policy decisions about maternal immunization research and programs. And perspectives of women who are or were recently pregnant should be considered while designing and implementing maternal immunization research. Now, moving on to the regulatory guidance for maternal immunization clinical research. We saw that, with COVID-19 vaccines, pregnant women were just not enrolled during the clinical research of these vaccines. And so when these COVID-19 vaccines were actually licensed for use in adult populations, the regulatory authorities did not have the data to make a recommendation for pregnant women to receive the COVID-19 vaccines initially. And so, one good thing that actually came about out of the pandemic was that now the stringent regulatory authorities have become really committed to advancing clinical trials in pregnant and lactating people. I'm just going to walk through the regulatory requirements of the FDA for maternal immunization research, because this has really been clearly spelled out in a number of draft guidance documents that have come out recently. But the EMA and other stringent regulatory authorities are well aligned with the FDA. In fact, the International Coalition of Medicines Regulatory Authorities, or the ICMRA, recently had a meeting, and there was complete alignment on the regulatory authorities being committed to advancing clinical trials in pregnant people. So the 10 requirements for FDA, the 10 requirements for the FDA for maternal immunization research include the following. So when scientifically appropriate, non-clinical studies, including in pregnant animals, and clinical studies, including studies in non-pregnant women, should have been conducted and provide risk information. There should be a prospect of direct benefit to the woman or fetus. And if there's no direct benefit, then the risk to the fetus must be minimal. The research should be required to develop important knowledge which cannot be obtained by other means. The least possible risk for achieving the objective should be done in, should be as part of the clinical trial. Informed consent must be obtained from the pregnant woman. If the prospect of direct benefit is solely for the fetus, then additional consent from the father is needed, unless he's unavailable, is incompetent, is temporarily incapacitated, or the pregnancy has results from rape or incest. The participants have to be fully informed of the reasonably foreseeable impact of research on the fetus or neonate. For adolescents who are pregnant, assent and permission needs to be obtained. There should be no inducement for pregnancy termination. The investigators of the clinical research should not be involved in decisions regarding pregnancy termination, and investigators should not be involved in determining the viability of a neonate. So these are really pragmatic and easy and implementable recommendations that the FDA has made for for maternal immunization clinical research to be conducted. Now, the prerequisite for inclusion of pregnant women in in the clinical trials includes there should be adequate non-clinical studies, which include the developmental and reproductive toxicology or DART studies which have been completed. And the clinical trial holds prospects of direct benefit to the pregnant woman and of fetus, which is not available outside the research setting or cannot be obtained by other means. The prerequisites for inclusion of pregnant women in post-marketing studies is that it should be adequate non-clinical studies have been performed showing that the vaccine is safe in animals. You should have an established safety database in non-pregnant women from clinical trials, or preliminary safety data from the medical literature or other sources regarding the use of the vaccine in pregnant women. And the efficacy and the safety cannot be assessed by other study methods. So the research is required and not something that that the results can be extrapolated from other studies. Now, women who become pregnant while enrolled in a clinical trial, you could be enrolling women with reproductive potential who are non-pregnant, but some of them might become pregnant during the course of a clinical trial because there might be contraceptive failure, for example, or they might not have used the contraceptives adequately. So unblinding should be done so that counseling is offered based on whether the fetus has been exposed to the investigational vaccine, placebo, or control. And the risks and benefits of continuing versus stopping the vaccine candidate should be reviewed with the pregnant woman. And the investigators have to determine whether the non-clinical data is adequate to support lack of risk, and the benefits of continuing participation of the pregnant woman in the research outweighs the risk. So then the woman, if a pregnant woman chooses to continue in the clinical trial, then a second informed consent process should be done that reflects the additional risk-benefit considerations, and reconsent is then done for the pregnant participant. And again, explaining the risks and benefits of participating in the research. And the pregnant women, of course, should be able to decline participation or additional follow-up at any time that they so choose. And regardless of whether a woman continues in the trial, it's important to collect and report on the pregnancy outcomes. So, we need to report on the maternal and neonatal outcomes for every woman who might become pregnant during the course of a study. In terms of timing of enrollment of pregnant women in clinical trials, so the recommendation is that the phase 1 and phase 2 clinical trials are done in non-pregnant women, including females with reproductive potential, and these trials should be completed before enrolling pregnant women in later stage clinical trials. And if there's limited safety data or other approved safe and effective treatments available for that disease condition, for example, then you could complete the phase 3 clinical trials in non-pregnant women before enrolling pregnant women. And if there's limited therapeutic options, the risk-benefit consideration may favor enrollment of pregnant women in earlier phase trials. And if there's safety data for a vaccine that's been studied previously for other indications of populations, the risk-benefit consideration may favor enrollment of pregnant women in earlier phase trials. For example, with COVID-19, early in the course of the pandemic, we did not have safe and effective treatment options available. So then following these updated guidelines, pregnant women actually could have been enrolled in the clinical trials at a much earlier stage than they were, than what we actually saw for the vaccines that were initially developed for COVID-19. In terms of minimal safety data collection when pregnant women are enrolled in a clinical trial, this includes the gestational age at enrollment needs to be collected, gestational timing and duration of vaccine exposure, pregnancy outcomes including adverse maternal, fetal, and neonatal events, and cord blood collection at the time of delivery. So this is just a minimum data that needs to be collected, and additional data, of course, can be collected as required for the trial. Now, coming to the safety evaluation of maternal immunization clinical research in LMICs. So safety of maternal immunization is a key consideration for all the stakeholders, including the pregnant women, her family members, healthcare providers, ethics committees and regulators, the researchers, vaccine manufacturers, public health programs, civil society. And the problem is that safety cannot be measured directly and can be only inferred from the relative absence of adverse events following immunization. And there's no inability to de-challenge. So unlike in a drug trial, where you can stop giving the vaccine, once the vaccine is administered, you cannot de-challenge and stop the administration of the vaccine, for example. In maternal immunization studies, the enrolled population of pregnant women is broad. They have no defining illness. The outcome might be uncommon, but serious. And efficacy is often defined as a biological rather than clinical response. So you need very large sample sizes for a number of, say, phase 3 clinical trials in pregnant women. And safety and efficacy has been demonstrated in the vaccine recipient and the infant to support the indication. And the subpopulations, including the pregnant women, neonates, and infants, say they might be more susceptible to adverse events in pregnancy. So targeted neonatal and infant ages may coincide with the emergence of underlying diseases. Like we said, there might be congenital malformations that occur as part of the normal background rates, but they might be seen when, the neonate is born. And if the background rates are not known in the country, then the research community might attribute those rates, those congenital malformations to the vaccine that's been given. So it's very important to know that adverse events are often ascribed to maternal immunization in LMICs. And safety assessment requires real-time assessment of risk versus benefit. There's limited availability of baseline data on disease burden and maternal outcomes during and after pregnancy. And this is required for the safety monitoring during trials and interpretation of data. So you need to distinguish between vaccine-related versus pregnancy-related outcomes. So it's not, even in low-risk pregnancies, we might see abortions or stillbirths occurring, or even neonatal deaths occurring. And so not every adverse event that occurs is due to a vaccine. So it's important to have a systematic approach to classify adverse events to be able to assess causality. So you could define adverse events as maternal or infant. For example, intrauterine growth restriction could be a maternal adverse event, while small for gestational age could be an infant adverse event. The severity scale for grading the severity of adverse outcomes should be based on pregnancy-specific physiological laboratory values. And the same scale should be applied throughout the clinical development programs so that you have consistent measurement that's used as a benchmark throughout the program. The halting criteria based on predefined thresholds for relevant events observed during the clinical trial should be decided well in advance. And you need validated developmental assessments for newborns to be consistently done during the course of the research. The other key challenges are that there's variable infrastructure, diagnostics, standard of care, and social and cultural norm that we see in LMICs. And the health professionals who recognize and report the adverse events are often not those who administer the vaccine. So the benefits of maternal immunization are not immediately visible, and there's a knowledge gap of early pregnancy vaccine response and safety. So most of the current knowledge for maternal immunization comes from observational studies conducted in the latter parts of pregnancy, and we have very limited data available for vaccine being administered in the first and early second trimester or from randomized clinical trials. And we have rudimentary pharmacovigilance systems for adverse events often seen in LMICs. And, the pharmacovigilance systems for maternal immunization are often much worse than those for EPI programs. So the introduction of new vaccines for maternal immunization will need to develop active surveillance systems for pregnant women who are vaccinated and their infants. In terms of passive surveillance, which is the pharmacovigilance, which is commonly seen in LMICs, this relies on assessments of spontaneous reports of adverse events following maternal immunization and literature case reports. The advantages are there's ease of implementation, low cost, and you can capture unexpected events. But the issue with passive surveillance for maternal immunization is that there's incomplete data on the pregnant women's genetic disposition, lifestyle factors, timing of exposure, birth outcomes, and additional risk factors that might have resulted in the adverse outcomes. There's lack of denominator and control group data. There might be low selective reporting of adverse events by healthcare providers in LMICs, recall bias associated with retrospective reporting. Poor case documentation and incomplete data on the adverse events is often seen, and lack of standardized case definitions to enable data comparability in most countries. So it may help to provide a signal for higher than expected adverse event rates or unrecognized adverse events, but you cannot use passive surveillance for determining if the rate of the adverse event has increased or if there's a causal relationship between the adverse event and the vaccine based on passive surveillance. For this, you would need active surveillance, which would be used for characterizing the adverse event profile, the rates and risk factors. There's logistical and resource constraints limit its wide application of active surveillance. So LMICs might carry out active AEFI surveillance only for selected AEFIs at selected institutions, which would be the sentinel sites. It need not be done across the country. Large linked databases are administrative bases with well-defined populations which are created separately from each other, and then they're linked to enable sharing of data across platforms, and you can detect rare adverse events using these large linked databases. But in LMICs, there's limited quality of information in these databases. So in LMICs, practically, you could use a network of hospitals or health centers within the country for active surveillance, using self-controlled case series to overcome the difficulty of obtaining denominator data to calculate the rates of adverse events. But the limitation of active surveillance is there's lack of harmonized methods of vaccine safety studies, lack of standardized definition, and causality assessment for adverse events. Pregnancy registries, especially prospective observational studies, may be vaccine or disease-based, and these are often conducted by manufacturers as part of a post-marketing commitment. The advantages of pregnancy registries are that you can collect information on exposure of pregnant women to various types of vaccines, or registries can capture specific type of diseases which would be of relevance to specific types of vaccines. A single registry could collect data on many pregnancy and infant outcomes. You could monitor for suspected risks raised by the trials or from case reports. You could identify factors that affect the risk of adverse outcomes, including the dose, timing of exposure, and maternal characteristics. The pregnancy registry should serve as a hypothesis generating tool, and you could get real-time data as soon as the vaccines become commercially available, unlike electronic health records where there's a lag time in data availability. So there's less exposure misclassification, recall bias, and confounding seen in pregnancy registries. In fact, that's one of the reasons why I'm leading the setting up of a pregnancy registry in one of the low-income countries in Africa, so that we would use the data from the pregnancy registry for future clinical research in these countries. You can get data from physical examination of newborns and periodic clinical assessment of infants of exposed mothers about the outcomes of interest. So generally, the limitations of the pregnancy registries are they lack denominator data. Generally, they're very small in size and they lack the power to detect the association for rare outcomes. Pregnant women recruitment and retention is often very challenging. And so then you have to have specific efforts for facilitating recruitment and retention, including partnerships with disease registries, linking to electronic health records. We're using tools for like mobile apps in some of the pregnancy registries we're working on, and we're using advanced social and behavioral science techniques for recruiting pregnant women. For the comparator group, we are recruiting within the study, we are recruiting unvaccinated, concurrently enrolled pregnant women in the pregnancy registry that we're working on, but you could also have external groups and background rates being used. And you might need to conduct case control and retrospective cohort studies to collaborate the findings from the pregnancy registry. So active surveillance for maternal immunization should be well integrated within the national healthcare system, and there should be integration with the existing maternal neonatal child health services and surveillance. We have to focus on building existing infrastructure and capacity and not setting up de novo systems. So 3 information categories which are common in LMICs include demographic surveillance systems, mobility and health outcomes registry, and pregnancy registries. So these all would need to be linked with unique identifiers and pregnant women and infant identifiers linked and integrated in an active vaccine safety surveillance system. And this will strengthen existing programs, leverage the infrastructure, and minimize disruption of routine MNCH services. So it's really important to use standardized case definitions for maternal and neonatal outcomes, enabling terms and standards for data collection. So this will lead to a common understanding of the key events related to immunization. There could be data comparability, merging of data sets, and consistent analysis of clinically important adverse events across clinical research. You could use the standardized case definitions for global safety summaries, rare adverse event determination, and for strengthening pharmacovigilance. This would increase the accuracy and precision and comparability of the data. And if you had ICD and MEDRA coded event for case identification and standard case definitions for case verification, this would help improve the data quality. So, we set up the GAIA, or the Global Alignment of Immunization Safety Assessment in Pregnancy. This was a network set up with a specific focus on maternal immunization safety, with a focus on LMIC needs. And, I was fortunate to help set up this GAIA network. The outcomes that we had were case definitions for 26 key obstetric neonatal outcomes, glossaries for well-defined terms like gestational age assessment, guidelines for data collection, analysis, and presentation, guidelines for collection of case report form variables, obstetric risk assessment tools. We had codings, including the MEDRA, ICD-10 and 11 codings, and case identification algorithms prepared. We set up working groups for developing these case definitions, which consisted of academia, public health organization, regulatory agency representatives, representatives from vaccine manufacturers. And the case definitions we set up had 3 levels of certainty, and these were case definitions for diagnostic certainty. So the definitions had a level 1 with sophisticated diagnostics, which was the gold standard and could be used in high-income countries. Level 2 of the case definition with less sophisticated diagnostics, and level 3 with simple diagnostics that could be used in a LMIC setting. So the definitions that we developed increased data accuracy, precision, and comparability. So these were the 26 neonatal and obstetric case definitions that we developed, and they are all published. And so the WHO Global Advisory Committee on Vaccine Safety provided a highly supportive assessment of these GAIA guidance documents and considered them to be timely and useful. And these definitions and guidance are now being utilized in clinical research, epidemiological studies, post-marketing surveillance, pregnancy registries, and post-licensure studies. And field testing and validation of many of the GAIA's case definitions have been done, including in both LMICs and high-income countries. So we've also now developed companion guides for key maternal immunization case definitions, where we've detailed the risk factors and background weights for the event, key caveats for the case definition for real-time investigation needed to meet the case definition, guidelines for analysis and presentation of the event as an adverse event, detailed case report forms that would guide the data abstraction and interpretation of the data. And I, worked on developing the companion guides for some of the key maternal immunization adverse events, essentially, including spontaneous abortion, preterm birth, and we're working on stillbirth. So now some of the other initiatives for maternal immunization safety. This was a program that I was fortunate to be part of, where we summarized the existing programs in maternal immunization pharmacovigilance and identified the gaps and needs and outlined a roadmap for program development and implementation for monitoring maternal immunization safety in LMICs. I was part of the PREVENT project, where we developed ethical guidance for inclusion of pregnant women in the development and deployment of vaccines against emerging epidemics. And WHO SAGE found the guidance to be very timely for the R&D for vaccines against emerging infectious diseases. And this guidance was then implemented for Ebola and COVID-19 vaccines. WHO is now working on a number of initiatives on safety surveillance in pregnancy. I'm part of the WHO Expert Steering Committee on Safety Surveillance in Pregnancy in LMICs, and we're helping to advise WHO. So this includes defining a minimal data set for monitoring maternal and newborn health, looking at the priority maternal and newborn health indicators, evaluating the safety of COVID-19 vaccines during pregnancy, looking at existing pregnancy exposure registries and identifying the gaps and recommendations on the strengths and limitations on these existing registries. And we are working on a WHO guidance for conducting pharmacovigilance in LMICs, including the definitions of key adverse terms, the minimal data elements that are required for pharmacovigilance, the methods and tools for data collection, among other elements in this guidance document. This will be available by Q1 2024. In addition, we are doing a lot of work, including on Group B strep vaccines for maternal immunization. I'm helping to develop the ECVP, or the evidence consideration for vaccine policy for Group B strep vaccines to be given in pregnant women, where we are defining the data that will be required for the regulatory and policy recommendations for these vaccines. We hope to define what should be the parameters involved in a Group B strep maternal immunization effectiveness study. So these are discussions that are ongoing. So this has just been to give you a snapshot of some of the activities that are being done for maternal immunization research. So in conclusion, conducting maternal immunization research in LMICs is challenging. There's limited availability of epidemiological data on disease burden, maternal neonatal outcomes commonly seen in pregnancy. There's lack of harmonization of standards for data collection, assessment, and analysis. There's various cultural beliefs and challenges in recruiting and retaining pregnant women in studies, challenges of obtaining informed consent, and variable infrastructure and logistic capacity seen. So now we actually have adequate regulatory guidance to address a number of these issues. The maternal immunization clinical research may be based on evaluation of the risks to benefit evaluation for the pregnant women and the fetus. So you have to evaluate the benefits of the vaccination versus the risk of leaving the disease untreated or treated with available treatment options. And it's always a risk-benefit analysis that the regulators look when they are licensing any vaccine. Early conduct of non-clinical studies or the DART studies in animals should be done so that this would allow timely participation of women in clinical trials. You need to tailor the recruitment, consent processes, and retention strategies. Use the maternal neonatal outcome definition, the data collection method that align with global efforts. It's very important to partner with the MNCH and ANC providers and community and local healthcare systems. So you have to strengthen the existing programs, leverage the infrastructure, and minimize disruption to the routine MNCH services. Learn from the best practices of conducting research in LMICs, especially with regards to the logistics, quality assurance, and training. So in introduction of new vaccines of maternal immunization will also require prospective, active surveillance of pregnant women who are vaccinated and the infants. And we are helping set up some of these pregnancy registries that would work for this active surveillance of, once the new vaccines like RSV and GBS are implemented in LMICs. So researchers need to be able to respond rapidly and thoughtfully for all eventualities, and maternal immunization, uh, uh, clinical research is always a multidisciplinary collaborative approach, especially if it's done in LMIC. Thank you so much. Now I would like to open it up for questions.